Thank you so much, Malvika, for your time. So today's speaker, first of all, I am Renuka's husband. Okay, so Renuka Suravajhala finished her master's from Kakatiya University, and then she obtained a PhD in synthetic biology, working for several years in Denmark and also in Singapore, Roskilde University, and several other places. Our research interests are largely in the areas of molecularly imprinted polymers for development of certain organoid models, for cancer cell lines. She has steadfastly worked on various cancers, including prostate cancer, and also development of certain long non-coding RNA docking models over the years. And she's also a co-founder of Bioclues and founded the Women in Biology Forum in Bioclues. She is she also won the prestigious Young Scientist Award from Microbiological Society of India, and also other things. She is currently a research scientist and assistant professor at Amrita Vishwa Vidyapeetham. So since we both share the same office, I'll be muted, I'm sorry, and I'll also put on these kind of things, just we might get an echo. We're in the same office, we share the same office in Amrita University. So this brings to you, Renuka. Thank you so much. Your screen is not visible. Can you please share your screen again? Yeah. So she will start about the introduction to docking, and there will be hands-on sessions, followed by at 8 o'clock, I'll probably take care of some kind of, hands-on, and then Sandeep will take over, during the last 15 minutes. Yes, now open the presentation. Yeah. Okay, now you can make it full screen, please. Thank you so much.
Thank you, Prashant, for giving wonderful introduction, and I'm happy here to be with you all, sharing my knowledge on what I have in molecular docking. Is my screen visible?
Yes, ma. Yes, ma.
Yes. Yes. So, just before going to molecular docking straight away, I just want to talk why we have to consider molecular docking in our studies, and how it helps in identifying the lead molecules. As you all know, we have various sources of drugs and molecules and chemical molecules in the form of drugs, which can have some kind of impact on our system. So these sources of drugs are from various sources, coming from the animals, plants, inorganic, synthetic materials, and all. And these have very importance in various factors, various mechanisms and pathways, in different ways. So in drug designing and discovery, we have the two phases. One is drug discovery and as well as the developmental phase. But in drug discovery, it plays a very important role, that how a drug can be designed and what helps us, what actually the structure of the molecule helps to target to a specific site, and also what are its properties and all. So these are part of the drug designing, and we can go with different in different ways, these drug designing. One is like by luck, we get so where we try to identify some molecules that have more efficiency to increase the activity of certain functions, then they are come into the serendipity. And other way is Based on the structure of a molecule, we build the libraries, which are nothing but the descriptors, molecular descriptors, and try to identify how the each and every molecule of these atom of the structure plays a very important, and where exactly we have to figure look in. So these are with respect to the structure based library screening. And another thing is, if we have a molecule, can we change or make many analogs for these molecules, and try to say how they actually increase or decrease the mechanism or the action. So in this way, the drug modifications also plays a very important role. Apart from that, rational drug designing, in which we have pool of molecules and then try to see how they can actually play, what are its properties, how, looking into the docking, molecular interaction studies with respect, we applying molecular docking, and also trying to understand the stability of the complexes with a protein, and then trying to go for a big process of identifying, and as well as going for the clinical studies, and so on. So the rational drug designing is also a way where we get approval from the FDA after the clinical studies. So this is the overview of the drug discovery phase, in which the primary discovery plays a very important role, and which target or the protein where we have to target, which site it is, and what is its role in the mechanism, how we have to actually look in. All these will come under the target identification and also target validation, where how many targets are there. There can be multiple targets as well for a specific action. So in that way, it helps us to identify what are the targets we have to look in, and how we can pool these targets as well. And later on, the drug discovery phase comes to the early safety and as well as lead optimization studies. So in an overview, we will try to if we have a protein, targeted protein, the sequence we get usually from the FASTQ sequence, and try to either if we have the structure of the sequence, then we don't need to do the ab initio homology modeling. Otherwise, if we don't have the structure of if the structure of the protein is unknown, then we have to go for the homology modeling or threading based approach or ab initio approach. So these are the three methods where we can design the structure of the protein, and as well as later on we can identify the pockets. This protein, if we have a structure of a protein, then automatically you can use the databases which are available in the EMBL and as well as in the RCSB database. It is a protein data bank is already available. You can find the structures of those proteins, but how we can take the structures, it is very important that which structure we are going to choose for our docking studies. That also very plays a very important role. Otherwise, if we have a pool of molecules, try to screen the properties, then we have to go for the pharmacophore modeling. And later on, once we screen the molecules and also identify what is the target and whether it is having structure or not, and if we don't have the structure, we'll go to do the homology modeling. If we have the structure, we will identify the residues, and later on go for the interaction studies, where there are several softwares, and also Python programming is also available, where we can do the docking studies, and commercial softwares are also available. But we will go today with the open source software, which is quite useful and also helps to have accurate, more accuracy. So, and structure be where to know that which kind of like docking studies we have to do. We should know that whether we what we know actually about the protein and what we don't know about the protein. Like if there is unknown proteins are there, then which kind of designing we have to select. And if the protein is known and as well as the target is molecules are known, then we will go for the ligand structure based designing, which is nothing but the protein ligand docking. If we know the structure of the protein ligand but not the protein, then we go for the QSR pharmacophore modeling, and as well as try to identify ligand based approaches. So, and if both if the structure of the protein we don't know, like we have to identify a pool of molecules from the skeleton of the molecule, and then have to do something with the known protein structure. Then we can also look into the de novo fragment based drug designing, so which also have some kind of information from the experimental data as well. So that is about the dream about drug designing. Just I will give the overview how we actually go with this, and here we go for this fragment based drug designing. And here we have a specific site in the protein and try to identify which kind of molecules can fit into these sites, and how they can block these specific site. So we try to find whether it is aromatic chain or the alkyl group or any group which can fit into the pocket. We try to look in and try to develop some analogs, build the analogs, and then go for the docking. So, how to get the molecular information? So, when we start working, we need to know that some basic information is very important to understand regarding the compounds which we are actually working on. So, Campbell is really good, and also PubChem is also good. So it gives the information about the literature which is presently available, and what are the published papers which are available, and how much kind of work it has been in, and are there any analogs with respect to the specific molecule which we are looking, and also the vendors where the pharmaceutical data is also available, and so on. Like, is it an FDA approved or still in the clinical trials or what? So on. So all this information you can get it from the Campbell, which is quite user friendly, and also in PubChem. But compared to both these, I would prefer to have more information Campbell. And PubChem also maintains all the information, data, everything. The only thing is there is how it looks, which kind of data they are going to get. So both are equally good. You can choose any of these databases to identify the molecules and also know the basic information about the molecules. So so that is about Campbell, and then directly I will just move on with the what is docking. Here we try to it's a lock and key mechanism, simple lock and key mechanism, but happens in our biological systems as well. But the receptor can be a macromolecule, ligand can be either small molecule or macromolecule. So both, if they are having the orientation, proper orientation structurally, then only they can fit in each other. Just like our lock, and if we have designed key proper, the same key, then only it will fit, and then only we can open the channel. So similarly here in the biological systems also, if we don't have a proper ligand to fit in into the specific receptor, then there is the action cannot be the same, and we cannot expect the same kind of action. So that is the reason why we try to look into the molecular docking and try to see where the actually the interactions are happening. So in coming to the various ways where molecular docking can be implemented is with respect to the protein ligand docking. Ligands where ligand can be flexible, it can go around the receptor because receptor is a macromolecule, and it will be rigid usually, so that we can find the pockets. And here, the conformational changes of the ligand plays a very important role to understand the interactions. Whereas in protein-protein docking, we have two rigid molecules which are macromolecular and try to see whether there is any possibility of conformations. But usually, we don't have a proper software for the protein-protein docking. We have several web servers which can be useful. So yeah, so this is how the molecular docking and the protein, which is a receptor, and the small molecules try to bind. And if we have to look into specific pockets where we have to dock, those are nothing but the binding sites. So what are binding sites? Which are nothing but the amino acids in the sequence of the protein, and where there is a possibility of binding. So each and every protein has its own binding pockets. So to identify what are the active binding sites in your protein, you can use various web tools here available. Even mm, yeah, so Caspi is quite useful, and as well as so as other web servers are also developed. They are also equally good, and you can give the FASTQ sequence of the protein and try to identify, or else even the PDB ID will also help us to identify the protein. So now I will try to give a simple FASTQ sequence here, which I will give little later in the technical session. So, coming to the binding sites identification, these are the hotspots where there is a possibility of hydrophobic surface interactions, and as well as hydrophobic hydrogen bonding sites as well. Other than that, there are other interactions also there. That is van der Waal interactions and electrostatic interactions, which also plays a very important role in the binding sites. But basically, we try the binding sites are focused towards the hydrogen bonding interactions, and that helps us to know much about the function, how they are interacting, and all polarity. So when we go for the screening of the virtual screening, then here we try to take the molecules and then identify the active sites, and then later on go for the docking. And what we identify in the docking studies is the score is nothing but where the how the conformations are, what is the ligand posture which is changing in a specific site, and what is its binding energy, which plays a very important role. And the least binding energy has the more binding affinity. So that is which we have to look in. And these are the various physics based algorithm binding functions, which plays a very important role to understand the basic binding functions, and how these scores are calculated, and which helps us to know that the binding scores are based upon the non-bonded and bonded hydrogen bonds, and also physiochemical interactions and as well as the electrostatic interactions, which helps us to come with the delta G value. So coming to the what are the softwares which are available for the protein ligand docking? I know that there are so many GitHubs are also developed, but the standard softwares are the Autodoc, which is open source, and which is based on this. There are so many commercial and softwares are also developed, like Gold, Flex, Glide, and also Molsoft Dock. So these are the various docking softwares which are available. But today I will be focusing on the standard software, which is nothing but the Autodock. And I have already shared in the protocol how, yeah. Any questions, please? Are you okay? Like, yes, please. You can unmute
Am I audible,
yourself
ma'am?
and then talk.
Am I audible?
Yes, please.
Okay. Ma'am, I want to ask that in protein ligand docking, is it always necessary that the protein or macro?
Hello.
Yeah.
Yes, please.
Okay. So my question is that in protein ligand docking, is it
Huh.
always in protein ligand docking, is it always necessary that protein should be rigid or it can also be flexible?
It can be it should be reset because the protein is the ligand will be flexible all the time, so that is the reason the protein should be rigid because we try to develop the parameters with respect to the grid is made for the protein, so that we whatever parameters we are giving, we are ensuring that it will not move around, because if both both ligand and also the protein moves around, then it is very difficult to come up with some kind of stability in the complex. So if we can if we put the protein stable and try to see the postures of the ligand, then it is because already protein is a complex molecule, so it is better always to have one rigid, which is nothing but the macro molecule, and try to go with the flexibility with the ligand.
Okay, thank you, ma'am.
Yeah. Any more? Okay. So the here I just want to give the overview of the docking, how we do the docking with this. Once we identify from both the chemical databases and as well as the sequence of the protein, and then take the structure of the protein in the form of PDB and also ligand, we have to transfer, because usually ligands will be in MOL 25 or in SDF format. So the format of the molecule should also be converted. I will tell you how to do it in our technical session. And once we do the conversion, then there is a phase where both the molecule, the small molecule receptor and as well as the ligand will be in the same format, PDB format. But Autodoc has the can set the parameters in the PDB-QT form, where we try to optimize some of the remove the water molecules, additional water molecules which are there, and then also the partial charges are added to the protein, and as well as per the ligand. So if for ligand we try to see what are the rotatory bonds, torsion, how many rotatable bonds are there, how flexible is the small molecule to go around and try to fit in, and we try to set the parameters in such a way. And for the protein, we remove the water molecules and try to add if any hydrogens, like for some residues, hydrogens are not there, then we try to add them, and then try to convert both the molecules, ligand and also protein, into PDB-QT format. So PDB-QT format is very important to understand the software that they will try to accept a PDB-QT format of the protein and as well as ligand. Once we take them, then there is some tab for the grid parameters. So in the grid tab, we will try to take both the ligand. What is the ligand? We are trying to make the system software understand. Okay, this is my protein and this is my ligand which I am looking in, and then can you set parameters for that, then it will give a little some few options how to set the grid. And in that grid we can grid is nothing but the 3D dimensions of the protein, where it should be, what xyz coordinates, how they have to be, and what are the limitations. There are some limitations to that as well, and which I will go in the technical way when we are doing it, and working with the tool. And there, once we do that, we will develop a file called GPF file, which is nothing but get grid parameter file. Similarly, once the grid parameter is done, there we will also do the docking parameter file, which helps us to how many dockings we are, means how many confirmations it has to go. If, because usually we don't accept anything until it has repeatedly it tries to bind in the same place about minimum, it gives around in the software 10 repetitions or 10 times, but usually we go for 25 time repetitions. So in that way, the docking parameters are also placed, where we try to take consider generic algorithm and Lamarckian algorithm as a standard parameters for selecting the docking, and try to develop this DPF file. So to run any file for docking, GPF grid parameter file and DPF file should be there in the same folder. So all these files which are generated during the process, like when we are getting the PDF PDB file, PDB-QT file, GPF file, all these files should always be there in the same folder. Ensure that they should not be scattered here and there, and then you are trying to pull all the files from each other. Then you will be messed up with so many copies, developing so many copies. So ensure that I will definitely request that you should make a folder and then try to start pulling all the files. So once these files are done, we need to either go with the docking, or with we can go with the command line as well. So I have already shared the protocol, and based upon your system compatibility, like usually Windows or Linux and Windows have two syntaxes which we have to do. One is with the auto grade, where you will do the auto grid command, giving the GPF file, and also output file will be in the form of grid log file, GLG file, which also creates maps. There are several maps which are developed based upon our 3D coordinates which we have given from the GPF file, and it will develop these maps for the ligand protein, where how it can go in and try to find where the possibility of interactions. And once the auto grid is run, it will take, depending upon the system capacity, it will take 1 to 2 minutes, and later on you can give the command autodoc command. Once in the autodoc command, you need to give the DPF file and then output file as DLG. So if you mention the DLG file as an, yeah, your output file, it will all the confirmations. I have made the program to run for 25 confirmations, though all these data will be in the in this DLG file. You can right click and open the file and then check what are your confirmations, whether it is able to read properly, is it stuck up in between, or it was able to process 25 runs. And then I will also show how to analyze the data, and also how to visualize the complexes. So these are the basic parameters which you have to do for the autodoc ligand preparation, and as well as the autodoc protein preparation. And this is where the both the PDB qt files, or we try to uh, develop them. And this is where we try to, as I was telling you before, the grid, the xyz coordinates are nothing but, here we have the xyz coordinates, and also the spacing, where how much resolution it plays a very important role. So all these are very important when we are doing, how we are saying the saving the GPF file. So this is how we do the grid parametering. And the outcome is nothing but the binding energies are calculated in the DLG file, and the algorithm or the software ensures that it takes both the van der Waals forces, hydrogen bonding, and electrostatic interactions, conformations, torsions, and also the solvation. All together, it will calculate how much binding energy is there for specific conformation. So likewise, 25 runs, it runs means 25 times it tries to calculate the binding energy. And this helps us to come up with what is the binding energy for this. And another advantage with autodoc is you can also know the IC50 values. So in autodoc you have the virtual screening as well, where most of them they try to use autodoc vena, where you will get the binding energies but not the IC50 values. But if you do autodoc 4, definitely you will have the information about IC50 values, which helps us whether we can, if we get a good candidate, how much concentration of the drug molecule we have to consider for in vitro studies. That is all, that information all is also can be generated from autodoc 4. So once we are done with the docking, we have to visualize. Means we need to know where the residues are interacting, how they are interacting, and what are the residues, how they are. So there are two ways to visualize it. One is with the 2D visualization, another one is 3D visualization. In 3D visualization, you will only have, you will see the structure and also the residues, and here you will be able to clearly see the structural information, like the where they are interacting. So that is leak plot. I would, and also there are various other visualizations are also there. Leak plot is good, and also Discovery Studio is there. Chimera, Pymol, Rasmol, all these are visualization softwares for better for publication purpose with good resolution, high resolution pictures, and very user friendly. That is nothing but the Chimera is it's a open source software, and also the Rasmol is also good. I definitely recommend that we can use any of them. And also we will, once we are done with the docking, I will show how to do with this visualization as well. Yeah. Any doubts or any questions?
Excuse me, madam.
Yes.
Um, yes. Means it is compulsory to remove the water molecule from the both of the structure, ligand and protein.
No,
Before.
usually we don't have in the ligand, but it will be there in the protein.
Okay. Thank you.
Yeah, welcome.
Ma'am, one more question I'm having.
Yes, please.
Uh, I have used Chimera X for the docking visualization. So, is it possible to produce a 2D structure like we used to do in the Discovery Studio using Chimera?
There are possibility with MD as well with Chimera, but the thing is, we need to look into those things. But homology modeling, I don't think that it is there in Chimera.
Okay.
Yeah. Any more questions, please?
Yes, I have a question.
Yes.
Okay, sometimes while doing docking, I find out that if I predict my binding site in orthodox binner, if I superimpose the grid box all over the protein, sometimes the ligand will leave the binding site I have labeled and dock to another place. How do
Oh,
I do it to make sure that it binds to that docking, or does it mean that the binding site differ according to ligand?
the
Thank
binding
you.
sites definitely differ from for ligand, but sometimes maybe when you are selecting the output files, ligand protein files, and clubbing together to form a complex from with the output, then maybe you might be it is going mismatch. That is the reason they are still not docked. If it is dark, then you will be able to visualize what are the interactions. If it is not dark, they will be definitely be apart from each other.
It's actually dock, but it's not in the place I have labeled. It doesn't dock at the binding site where I have labeled.
Okay, maybe there is no possible interactions. That can be also a chance.
Then, is this all these league prods and Rasmore, are they open access,
Yes.
freeware like the
Yes,
camera?
yes, yes. Leak plot. Actually, you need to, if you have a academic email ID, then it is free, and you need to register. And every year they try to update the software, so automatically you have to ask for updation as well, like a request for extension.
It's all right. Thank you.
Yeah. Any other questions? Okay. So I will go with uh, from the basic, like how we have to convert molecules and all. So if everyone is okay, like moving ahead, I will start. So yeah, so I just start with the curcumin and I'm sorry. Yeah, I think I forgot something to tell as well. So yeah, so here in biochemical packages, we can also do with Python, and there are various various web clients where the web port stacks are also there, and then you can use them. And apart from that, this is how we have to look in, like if we have the sequence, try to identify whether the structure is not there, whether it is there, and identify the active sites, and also with the ligand, if it is a phytochemical, if you have reference, that is always good. Sometimes it helps us whether our molecule is good enough compared to the already available active molecules, lead molecules. If we have more efficiency compared to that of the available, then the control always helps us to come up with some kind of conclusions. So in that way, if we try to do the docking with the grid parameters and docking parameters, and then do the processing with the docking, and then the once we are done with the complex docking, then the visualization will be either in the form of 2D visualization. Here you can see the where the residues are interacting, and as well as in the complex visualization with camera, you will be able to see where exactly the your control and as well as the target lead molecule is also interacting, where it is binding. So that way it will help us to come to understanding that where the in the protein, where the weight is trying to interact and what is its role. Till that extent it is fine finding the residues, but later on the dynamic simulation will help us to understand the stability of the complex. So once we are done with the docking studies, it is always important that to know the stability of the complexes. So that is also very important, uh, right? So yeah. So there are various softwares available with the databases like Campbell. There are other databases are also available. You can, I will share this, and then if you want to use for a better approach, then you can always use them. So before I stop here, I will just go with the tutorials technical session, where here I have given the chemical molecule. You can give the structure of the molecule, and also here you have the lot of information with respect to the molecules which you have to choose, and also each of these have a specific id, so that you can choose. And even in the pumpkin you will be able to see the same. And here you have different ways where you can download the structure of the molecule, either in the form of embo mol file, or canonical smileys, or different forms are there. You can choose any of them, and then I will just select the canonical smileys here, and then go with the tool I have given. OpenBabel, it is also having a software. You can use it, but if you are a frequent user, otherwise you can simply try to use the cheminfo.org, where they have also used and adopted OpenBabel. Just input your smileys here, in which format the input is there, and then you can choose different formats here. That is nothing but the uh, mm, which format it is. You can go for the PDB QT format as well, but I will definitely go for PDB format only. Why? Because there can be a chance that some errors can also happen. So that is also very important that we should mm, uh, yeah, we should go with the 3D in 3D more coordinates and then convert. Okay, and then once it is done, you can click here and then download it as where I have to download. But I already have a uh, test here. It's already there, but I will definitely give as okay. So Curcumin 3DB file, and here I'm also using 3DB file. So this is how we convert from smileys to PDB file, and then once it is done, then our protein has PDB file which can be easily accessible with the software. And similarly for the protein as well, how to screen the protein, right? I'm not sure which protein is available. Okay. Have you downloaded the softwares which I have given? I'm really sorry that I have given quite a little late, but hope you have downloaded the software. Okay, okay, fine. Thank you. So here we try to go and see the structure of the protein, and I have selected 6K7G, which is a protein, membrane protein present in Homo sapiens. And if I see the sequence, then even I can download the FASTQ sequence as well, and I will just download the FASTQ sequence of yeah. Here I have the FASTQ sequence, and just trying to take the same one, and then I will go with the editor, right? Here I have the sequence. I'm just selecting it and going back to the blast, and want to check whether the structure which I have taken is okay. Like, is it good enough, or should I go with another structure? Is there better structure than this? So if I give the FASTQ sequence, go for the next, and then go for the blast, and then it will help me out to which structure I'm going to choose. Why? Because this plays very important role in knowing are there any missing residues in the sequence when the structure has been characterized. So that is very important, and which if you see here, the coverage area is if you see the coverage area in this structure 7K 6K 7G, it has 100% coverage, and others it has small residues which are like missing. if we you can select them and try to see what are the residues which are missing, and few residues are missing in the front. So that is the reason it is good that we know the alignment, and also see where exactly the residues are and what is missing in our sequence. So these some of the residues are missing. So this is very important when we are selecting the structure of any protein. So in here also we have some residues which are missing, and here the whole coverage 100%, all the residues are there, but not in the other two structures 7OHG also I can select, but some residues are missing. So that is the reason it was showing 96%. So always it is very important when you are going to go and do the target any protein you are going to select, it is better that you try to identify which organism the protein is characterized, and also which method they have used. Usually Cairo electron microscopy, instead of electron microscopy we can go for X-ray crystallography as well. So X-ray crystallography visualization has a better resolution, and also it has so that also very important. Here the method they have used for electron microscopy, is it electron microscopy or X-ray diffraction method, and what is its resolution? It is 3.30. It's quite high actually. If I have to tell, if I have to select, if there is any alternative, that is the reason. If you have 100% coverage, and then the next option will be like, if both the both the structures are having 100% coverage, then you have to go for the resolution and experimental data which has been available in the protein database. Then if the resolution is one is high and another one is having low, try to prefer the structure which is having low resolution. 2.2, 2.5 is the maximum resolution, but it also depends upon the complexity of the protein. So that is also very important. And also you can also see what is the length of the sequence, and also which how many chains are there mutations, are there any metalloproteins which are there any metals which are attached to it, and what are the ligands. Sometimes there are some structures or proteins which have the drug molecules also inserted. So nag is nothing but some molecule. If you want to target this location, then you have to remove this um, uh, 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 and then try to find where exactly it is located. If you remove that and target the same location also, that is also possible. So that in that way, that helps us to go for the specific binding. So that is where you can go for the specific target binding, and remove any drug molecule, and then try to focus where exactly these things are. Then you will be able to see that, okay, you one 185 it shows, so it is interacting there. Okay, so what is this? So in this way you can try to look in and try to check where I have to actually what is this molecule. It is glucose, glycine, 184. So in that way it will be good to know where the interactions are, and you can remove them in the MGL tool which we are going to use. So this is about how to choose the protein molecule, and then you can download the structure of this molecule as PDB file. So here you have the PDB file. You can download it and then use it. Any questions, please?
Ma'am, yeah, I have a question. While choosing the target sequence, we have to first take into account the percentage coverage. Uh,
Yes.
if we didn't found the 100% similarity, then we can go for the maximum percentage,
Yes,
right? And
yes,
after that we
yes,
have to select the resolution, that is minimum resolution or high. I'm confused means
yes.
if it is 2.7 or it's 3.1, so we have to choose which one from them.
2.5. The low, the better. The lowest, the
Okay,
better.
okay.
Any questions? Okay.
Excuse me, do you say the lower the Armstrong the better, or the
Yes.
higher the better?
And the lower, the better because it is compactly packed.
Okay, thank you.
Yeah. So, yeah. Now we are done with this, and then we will go for the Autodoc. So once you open the Autodoc zip portal, then you will be able to see the various ways you can install it. One is with respect to Linux and Mac users are also able to use, and Windows it is also available. So you can click it and then start the installation. Apart from that, there is another important tool which is also important with this is the Autodoc tool edit, which you have to click here, which is a graphical user interface where it helps us to do all the parameters. So once you do the this, you have to download it and then go to downloads, and then you will be able to download MGL tool. So MGL tool is the one which is a graphical user interface tool which helps us to do those parameter settings and everything. So that is in different versions. You can download them and then install them and open. So once you have installed MGL tool and also the Autodoc, Autodoc is to run the once you make the grid and doc files parameters ready, then you, to run the program, then you need the Autodoc, and this MGL tool is mostly used both for setting the parameters and also for running the docking.
Ma'am,
So
these two tools are same, ADT and MGL tools.
no, from EDTA it will go to the MGL tool. So ADT is nothing but the MGL tool only. Okay.
Okay.
Any questions? So once I have installed, I have the NGL tool in this format 1.157 and as well as Autodoc 4. So both these files are very important. It has Autodoc Vina and also uh, the AutoGrid 4.exe and also Autodoc 4.exe. So these two files are available, and then here Autodoc Vina is also there, but I am not going to run Autodoc Vina. Okay, so that is how it is. And then MGL tool. Here in MGL tool, uh, you if you are a Windows user, you will have a shortcut icon which you can click and automatically use. I'm an Linux user, so I have to go with ADT here it is, and then I will just type in my terminal as dot hash ADT. So it automatically runs, and then the tool will open. I hope it is clear till here.
Yes, ma'am. It's clear.
So yeah, so I'm done with the opening the tool, which is the ADT tool, Autodoc tool, and here I have saved two files which I want to use for docking. Nothing but the protein and as well as the yeah. So here I'm using both the protein ligand and as well as the protein PDB 6K7G in the form of PDB file. So once you have both the PDB and PDB of the ligand and protein, then we have to convert into PDB QT file of PDB QT curcumin ligand molecule and also for the protein. You cannot do it together, you have to do it separately. So I have selected this file which have generated from the smileys to pdb file, and then I am trying to convert as pdb qt file. So this is the file, this is the file of the ligand molecule I have selected, and I will go with trying to find what are the torsions, okay, and choose the molecule. It's already there, so I'm selecting it, and then it will tell me what are the rotatory bonds present and how much polarity is there, and also the torsion. So it helps me out to identify these molecule, and then I will try to save the and also I can also look into the aromatic carbons as well, but automatically I want to save as pdbqt file. So I will try to save as pdbqt file in the same folder where I'm working. So that is so I have created here for here, right, PDB Qt file. So this is PDB Qt file which I'm going to save it. So save it. Once it is done, remove the file by while right clicking it. Are you able to open your MGL tool? Can you please open and try if it is possible? Should I wait or continue? Please let me know if you start to work or should I need to continue. Should I need to wait for some more time?
Can wait for 2 minutes.
Haha. Yes, please.
Ma'am, we are installing this file.
Yes, ma'am. Yes, ma'am. Go
Okay.
smoothly.
So I will just continue the session because it will take long time to do. So once it is done, maybe you can try, and then we can also discuss in the WhatsApp chat. Hope that is good, right? What do you say? Have you started opening the tool, ADT tool, or how it is?
Yes, and we opened the ADT tools. And one more thing, uh, you have directly downloaded the PDB file from the PDB site, or you
Yes, 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 yes.
converted it to.
Is it the ligand molecule you are talking about or a protein? I have not done the protein yet.
Ma'am, protein.
Protein you can download
Ma'am,
from
protein
the,
molecule. I'm talking about.
you can download directly.
But we have to look it for the chains also. No, means we have to select only one chain or
No
something
need,
that you
no need,
you're
no
talking
need,
about.
no
So
need, no need.
we can go with the whole protein.
Yes,
In case
download.
of homodimer also, we can go for.
Yes, yes. You just download the PDB file or directly from the database. So once, yeah, I have selected the protein of the PDB file alone. Okay. Are you done?
Madam, for Windows, Autodoc means is installed directly, or means means
Yes,
in
yes.
my system it is shown it is installed, but it is not found anywhere when I search. So
Oh, there will be an icon, I think.
okay, okay, okay,
It will be in this form, Autodoc.
okay.
You will get an icon. Yeah. Okay. So you are in the process of installation, I guess. Most of you. Okay. So I'm just continuing because it will take some time for me to go.
I have installed both the
Okay.
software. Now I'm preparing ligand file.
Okay. So should I wait for 5 minutes more or how it is?
And you can select any protein file or the specific that you have chosen.
Any file, anything which just I'm just doing because to show you. So any file of your interest, that does anything which will be fine.
And can you just write the protein PDB
Yeah.
ID name in the chat box?
Yes, please. Okay. I will resume back because we need to work out so many things which with the time limit constraint I have to move forward. So once the protein I have loaded the protein. Now in the Autodoc tool I have loaded the protein, and then here we can find what are the chains which are present in this protein, and also what are the residues. So if there is anything relevant when you are doing the when you do the active site search, then you know that what are the residues which have a role in some specific action or pathway mechanism, then you can automatically select them as well. So why just you will have the chains which are available, click on which chain the residues are. If you just click, it will expand, and then you can select any residue which is your target. So okay, I need these residues in this pocket, and I am looking into this whether it can dock at this location. Okay. And similarly, in another chain, I can also select what are the. So if I see these are all nothing but the already existing residues, means ligand molecules which are attached to it. Where they are attached, I can see them. I can check them where they are bind to. See, here it is. So this is nothing but an active pocket. Here I can see where it is, and also another molecule of this. So here it helps me out. This pocket is very important for active site for some kind of molecules are already attached, which we can remove them and try to select the same residues and try to use them for selective binding studies. So that is about the selectivity, and you can select them and save them as PDB-QT file as well. So I'm not doing that because I want to go for the randomized docking instead of selective docking. So here, once I have loaded it, I have to edit the bonds and also delete some molecules which are available. Like some water molecules will always be there, but this molecule thing is there. So water molecules should be deleted. In the edit button, you have the option for remove water molecules, and then next to that there is an option for adding charges. So in this we need to add charges to the protein as well. So it helps us to make the protein unstable. So when it is unstable, then only there is a possibility of there is a possibility of trying to interact. So when to make the protein unstable, we are adding Coleman charges. So the Coleman charges are added to the protein, and then see that whether any hydrogen bonds are there, and if I have to add hydrogen bonds, is it polar only or hydrogen bonds? Usually I go with the polar hydrogen bonds. Whether any polarity is there, then it will be showing that. So they once it is done, and if you see, there are a lot of hydrogen bonds which are attached now, and it is almost prepared, right? Then once it is ready, then save it as pdb-qt file. So save as pdb-qt file. So where I'm going to save it. So here I'm going to save as pdb. I don't want to save in the same format, but okay. So that is pdb-qt file of the protein. Okay.
Ma'am, what is QT?
Qt files are the partial charged files of the protein, and as well as for the ligand. So once the PDB-QT file is developed, generated, we will remove the PDB files. I hope it is fine. Any doubts? Any questions? Okay. So I have two files ready now. One is the protein, and as well as for the, it's not generated. Okay. I just have to do it again, I think. Yeah.
Ma'am, can you also share the
Hello. I'm
the
not.
active site talking.
Hello.
Uh,
Hello. I think yeah, I was not able to hear you properly. Can you please repeat? Thank you.
ma'am, I'm asking, can you also show the active side docking
Yeah, active sites. Okay.
means apart from this blind docking,
Yeah. Here we have the CASP, which you can use for.
if possible?
Yes, yes. I'm just showing it.
Yes, ma'am.
I'm trying to use. Okay. Here you can give the PDB ID of any protein, and then it will give the interactions. So here, uh, so I just gave the PDB ID, and it is trying to give me the information regarding the pockets, and also where exactly it is going to find it is going to bind. Just giving, I don't know why it is not recognizing this protein, but I have another SARS-CoV protein, nucleocapsid protein, so I just thought that I'll just try with this. So sometimes few proteins it is not able to find, but usually it will find. Here also in Pro site, you can give the sequence and you will be able to get the information. And for that, you need to know the sequence. You can give the sequence, and we already have the FASTQ sequence. So just open the FASTQ sequence, select, and then simply type here and then scan. So ProSite will also help us to find where the active sites are and where you can target. And here also it should, but I'm not sure why it is not able to recognize. But 60, or you can try with another protein which is of your interest. That can be also possible. So in ProSite, we can scan the protein and try to identify where the residues are, and how we can check whether which pockets are there. So this helps us here. It will say that here in this region, like 414 to 420, there is a possibility of sites which are having role in ATP is phosphorylation site. So this location we can look in and try to check that okay it is also labeling here. So this site you can try to check in your protein where they are located, and then look into that specific site. I hope it is clear, but it will also work, but try to check with some other protein. Okay, I hope it is clear.
Yes, ma'am.
Madam, it is showing error when we are saving this PDBQT. I don't know why.
Mm,
Like earlier, it was seen in your step, no? One of the step. Then I think you're
yes,
correct.
yes,
No.
yes, yes. So it should actually write as PDB file. So it should write actually.
Yes, same
Okay.
process.
So these, I think there is some charge issue is there in the field. So I think it is not able to recognize, or some charges are not Coleman charges. Okay. Now cannot create all my just wait. Oh, I forgot to add assign ADT AD4 type. So yeah, I have to add the AD4 as well. So add AD4 to this. So if you add here, you have the option to add atoms with assigned radii, where it should be assigned as AD4 type. So once it is done, then only it will be able to save as. So now it is charged. Okay, is it okay?
Yes, will do accordingly.
Yeah, just add ADT, assign atoms, and then the charge is assigned, and then it will automatically take the molecules. So now I have taken both the created both the PDBQT file of the protein and ligand molecule. So I will just load both the molecules now. Before I have done single molecule, making the generating the single molecule, but now I'm ready with the both the molecules. So when they are not docked, usually they are far away from each other. But once we do, the postures will also come into picture. So this is about the protein and as well as ligand. And now we will go to the if you have done for a molecule, specific protein with a with a ligand molecule, and you want to do for a number of ligand molecules with the same GPF file with same grid parameters, you can open the file which has been created for one ligand, and you can use the same file, GPF file, for other ligands also, so that they have the same parameters. Okay, for multiple ligands, you can use for the same protein, you can use the open GPF file. But now I'm trying to choose the macromolecule. Macromolecule is nothing but the receptor or the protein molecule. So you have to select the protein molecule. And so it's already partially charged. So yes, it is charged. So do you want to preserve the charges instead of it any? Yes. So it's already there. So the charges is there, and then it is saying that these are the correct charges of it. Okay. Okay. Done. So it is processing, and then now we will choose set map for ligand. So once we set map for ligand, then it will choose based upon what is the ligand, and then what happened? Okay. Yeah. So once I have opened the grid box, then I have the option here as XYZ dimensions, which are nothing but the grid box of this protein. And I hope you are able to see, right? There is a box in between. So here you can also try to set that so that it has a good coverage, and where how much it has to cover, and where it has to, the interaction should be. So the maximum you can go for this is about one space angstrom. So it is better that go with little less, and also the space should not be like too much, so that it goes the protein goes or the ligand grows freely, goes around, but it should have limited space to find the residues. So it is always important that how we are going to set the grid, and what exactly how narrow it is, how compact it is, and are there any residues where are missing, and all the leaving, if any of the residues are missing, or what. So this helps us to design the grid of the protein. And then here, once it is done, you can also make some changes where the center should be like here for x coordinates, y, z coordinates, the center. If it is little up and you want to move the box, then you can do here, and little more possibilities to go with the center where exactly it is, how, whether you want to go move little bit, how it can be. So these parameters you can set based upon what you are looking at, but don't create too much space. Like if my protein is in this region and I'm giving too much space here, then you are giving more flexibility for the ligand, but try to be less space good enough for the protein to identify specific sites where it can bind. So save, close save current saving, and then I have closed. So once it is done, then I will be able to save as GPF file. So GPF file will be created as for mm, mm, I want. So just a minute here. I'm going to make it as this. So save as test.jpf, and then once it is done, any doubts?
Madam, what actually you have added means from that edit
Which one?
means
Here,
earlier when error was means coming, no? At that time you have added something means from edit.
edit atoms, assign AD4 type.
Okay, okay.
Okay. So this is about the grid parameters. Next comes the docking parameters. So docking parameters, you can select rigid or flexible, but usually we try to put rigid only. For I have told what might be the best way, like go for the rigid one, and then you can. I think it is already there. Okay. So I'm just selecting the protein, and then also the choose the molecule. Here I'm choosing ligand, right? I need to. I don't know why it is. Let me check. So once it is done, then we will go for the search genetic algorithm. Here you have the GA runs. This is the poses which are like standard. It will give us 10 runs, but we can go for 25 runs here. So here you can change the parameters. So here you can give us 25 runs, and rest of the things you don't need to change much here because these are all automatically set parameters which take from the genetic information which is available, and that is where the algorithm is developed. And once it is done, then yeah. So we have selected that, and then the output file, how it should be Limatian GA file, and then you will have the option of file of which type. So that is nothing but the DPF file, uh, which is yeah. Okay, one. Have some issues with my protein as well. Let me delete and then open. I just select this one instead. And then choose. Um, I have this something. I'm sure whether it is working. Let me check. So I think some changes are there. Usually it used to be there, but there is some changes which are there in the software as well. So it is not giving the set some changes which are also not allowing me with the parameters which I have set. Maybe the torsion, yeah, here the core, everything is fine, but yeah. So once you are done with that, and then you have selected the rigid file as the protein, and also the ligand file. I have selected the test protein curcumin here, and then it is showing me the DP ligand parameters with the torsion and all. Before we never used to get this, and it is a new update for this with dihedral and all. So this is you need to accept it, and then yeah, as I said, like a generic genetic algorithm which we have to which I have already said, and then later on develop as the DPF file with the markers where you can give us test. So test.dpf file. So once it is done, then the DPF file is saved, and you can run here as well the grid file and as well as the doc file. So for the grid file here, you need to give where the grid file is, like the path, program path, where you can select where the grid file is. So my grid file is here, so I will tell that the grid file is here, open, and then also I should tell that what is the file name, where exactly where the file parameter file is. The parameter file is just a minute, is here. So test gpf, that is fine. So I'll just select the same and then test GPU, and then the log file, which is nothing but test GLG file. So I have just selected this one, and I have already given the PDB of the I have already shared with you all the protocol. So yeah. So the same protocol you can, which I have shared with you, you can the same thing I have made the information ready for you, so you can use it. And here it is there. So for command line, if you have to do, then you have to use the closed columns, and as well as autodoc4.exe. And remember that the file where the program is running should have both the Autodoc 4 and Autodoc file. So both these files are not there in the same folder, then it will not run. So ensure that these two files should be there in the folder where you are running. So that is one thing. And another thing is like yeah. So this is the command for the Windows people who are working on, and this is the command line for the Linux users. So you can generate the glg file and also autodoc file in where the grid 4/p, and ensure that there should be space in between. If you give too much space or the space no space also, that is also it will not recognize actually, and then it will keep on showing you error. So ensure that the spaces makes lot of difference when you are running. So that is one thing which you have to ensure when you are running it. And I have changed the uh, play location of the grid. So yeah, so now I'm just launching and it is running. So if I don't have any errors, then it will automatically run, and I can check in my folder whether it is the test glg is developed. So it's developed, so I'm able to get the glg file. And then once it is done, then we will go with the dpf file. So once this dpf file I have to use for doing the DPF thing. Right? So here I can run the DPF thing, and for that also I need to give where the autodoc is. So autodoc is here, I am selecting here, and here I can select that where the file is. Okay, file is here, and the log file which should be generated is nothing but the doc log file, which is test curcumin. So now it will launch, and then once it is launched, I should get the output as DLG file. I think my system is
Excuse me,
quite
ma'am.
fast. Yes.
Then how can we get these two files, Autodoc 4 and AutoGrid? These two files, which is mandatory for running.
When you are downloading, as I said before, when you are downloading, the NGL tool is for making all these parameters, right? And another tool which you are going to download in zip is nothing but the Autodoc. So these two files are important to run this program. So this is Autodoc. So Autodoc 4, here you have the files. And I went to the graphical user interface, that is nothing but NGL tool. So NGL tool and Autodoc, both should be there to run this docking.
But ma'am, it's not showing in the folder where I have downloaded both these software, Autodoc and MGL2. These two files are
You,
not showing there.
where is it, your Windows or
It's window, ma'am. Windows.
it's Windows, right? Usually go to the C drive, and then you can see MGL tools separately. And for Autodoc you can see as SIRP, something of that folder. A SIRP edu or some folder will be created. In that, Autodoc and Autodoc VNAM both will be there. Check that.
And can you send them at the WhatsApp group so that we can directly download
Which one?
these two files,
I have
Autodoc 4
already
and
done.
AutoGrid?
Yeah, I have already shared.
I think you shared the link. I'm just asking about those two files. Yes.
Okay. Yeah. So once it is done, there is some error in mapping, but you can try to do it. I'll just share the DLG file of the previously docked. So this is how the once you have done the docking, then you will have a file in this way. Once you have run the two commands, then you will generate. If it is a error is there, then it will say the fatal error is there in the grid points and it is unsuccessful. That is because your grid is not proper and you have to set the grid. So try to go back again, make the grid properly enough, good enough, and then you can do the docking in a way. But I have done previously, which I can show you at this point of time because I'm running short of time. And here you will have the data and all the information regarding the grid parameters, xyz coordinates what you have given, which helps you when you are writing in methods and methodology what kind of xyz coordinates you have given and what is the grid point space which you have to give. And also there are water a lot of input files also be there. But apart from that, if you go little bit down, uh, so once I go down, it is a clustering histogram I just want to see. And then it has run 22 runs. And then it is telling me which one is the one which is having the least. So I go down and then find little bit more information that what is the run which is having the good pose and which ranks first in this. So the lowest binding energy, what is the lowest binding energy for this pose? All this information it will give, and it says that fifth run is the best one out of the 25 runs which we have run. And then out of that it calculates the estimates what is the free binding energy is minus 2.44 kilocalories based upon the outbound inbound electrostatic and van der Waals forces. And also it also gives us the inhibition constant ki value, which is nothing but how much is enough for this molecule to be active with to show this kind of binding. And this information you will not be able to get in autodoc vena. So that is a little that's the reason which autodoc is considered as the standard tool. And it can allow only one protein and one ligand at a time. But now there are so many updates in Python programming also. You can use the Autodoc as well in Python programming so that multiple ligands can be docked at a time. And the commercial softwares have adopted Autodoc in such a way that it will form. So once that is done, once we have the output, then we generate the complex PDB file. Complex PDB file, how we get it as PDB QT. So I'm just removing both of them. But how to do that, I will show you. Delete. We are going to the analyzing the results, because I'm running short of time, so I cannot think of waiting for a long time, right? So I have here the output file. Just I'm taking the protein pdb-qt file, okay, and then open. For analyzing as well, we need to choose the PDB-QT file of the protein which we are going to find. And here I have to choose the DLG file. So DLG file, this is showing error to me, but I have already done with the protein, with the same protein. So I'm using the same DLG file. Once I loaded that, I will be able to see what are the conformations play according to the rank energies. So I can check the energies and then try to see, and here I can save the complex as pdb-qt file. So I don't think that choose. Okay. So now it is able to allow me to save as PDB-QT file, not as PDB file. So again, I have to save the complex as well in the PDB-QT file only. So that I don't want to save now, because already I have it. And then once it is saved, I will remove both the protein and as well as all the molecules. Just open the pdb-qt file, which is having the pose of ligand and also protein together. So I will use this, and then here it is having both of them, both the protein and as well as ligand. So this is nothing but the extra one, which is ligand. So where it is, so here it can be, we are able to view where it is. So here it is. So that is one thing. And then I have to again convert these two. Now any visualization software can only read pdb files, not the pdb-qt files. pdb-qt files are only read by the autodoc tool. So that is the reason we are converting into pdb-qt. And now we have to again convert into pdb file. So we can save as pdb file, and I have already saved, so I don't want to save it again. Okay. So that now here, once you save as pdb file, you don't need any more autodoc tool. Now the two visualizations which I was sharing with you. One is ligplot 2D visualization. Okay. And here I'm sorry, I have not shared how to download it. So you have the option to open ligplot and then go for the installation. It will ask for registration, and then you can request, and automatically it will be downloaded. I already have it, so I don't want to go for that. And then here you will have a folder which allows you to have lig plus. So click on that, automatically it opens. Now you can open for 2D visualization and as well as 3D visualization. I'm just waiting for the liquors to work, but mm, I haven't. It has been quite a long time, but just see that liquors can be open. Uh, yeah. So another thing which I want to show is chimera here. I'm not sure why it is not working now, but usually it should work. Uh, Here I have converted to the complex PDB file from PDB-QT to PDB file. Now I try to open that file, and then here I can have the 3D visualization of the protein and as well as the ligand molecule. So here I can do lot of things with this, and I can select which one I have to select. All these will depend upon I can select any of the molecules and how it should be. Usually the publication wise, it will be for saving these visualizations. It's always good to have a white background. And also here we can see that there are different chains, and you can select each of the chains, color them whichever you like. In that you can go to the option of colors, and then chain actions, and then how it should be, whether you want to change the ribbon or colors or something. Then I want to change the color, then I can go for the actions. So similarly here you can change the colors of each, even for the protein, even for the ligand molecule also, how it should be. Like is it a ball and stick? It should be, and what should be the color? All these parameters you can set, and you can do that. And you can even go for the labeling as well, like label the residues whichever you want. Like if I want to label, then I can say like here you have the command line as well. Label residues, what I have to label, and specifier which one, how it should be labeled. Sorry. So here you have some more options to do the visualization, finding the hydrogen bonds, and also doing what are the electrostatic interactions present, and where they are, and how are they intramolecular, or what is the bond distance. All these things you can do the flexibility, and also there is a possibility of finding the interactions between the molecules. So before going for that, I need to choose the molecule. Sorry. And then I have already choose the protein, so I don't need to. And only hydrogen bonds, and I can also change the Armstrongs to 2 Armstrongs relaxation. So that is also possible. And you can I can also check whether it is inter or intra. So that is also possible, and then I apply on this, and I think there are not too many. I can increase the Armstrong so that I will have better interactions. Even I can select the control and then go for the labels and then say like how I have to name them. So here I can label, and then if the color of the label is also different, then I can also change it so that it will be identified and it can be labeled. But for the protein, for the interactions, I need to select the protein ligand molecule and then go for the binding interactions, hydrogen bonds. No, select at least one, and then yeah, I'm just finding up.
So
So in this way, you can find the interactions here. We can find the interactions, and also we can also see there are interactions very closely. You can see the interactions, and also we can label the bond distance as well. So that is also possible. So just play around with this, and you will be able to see. Okay. And I can select them and then label those residues where it is interacting. I hope it is clear, and you try to play around with the visualization tools, and you will try to understand why we are doing and what we are trying to come up with these docking studies. Yeah. With this, I will stop here. Thank you.
thank you so much, Renuka, for your time. Friends, I hope you could have a gist of molecular docking. Uh, so let's thank Renuka with a big round of applause. Thank you so much, Shivy. She's in the group, so you can always ask any kind of questions. Please practice those kind of questions, and